Hello Nana, welcome to our channel KR Microbiology classes. Today the topic we are going to discuss is tuberculosis, which is one of the airborne diseases caused by the bacteria Mycobacterium tuberculosis. In this topic we will discuss about the morphology of the bacteria, cultural characteristics, pathogenesis, laboratory diagnosis and everything. Okay. So first we will start with the morphology of the bacteria. Mycobacterium tuberculosis. It belongs to the family Mycobacteriaceae, and this tuberculosis disease is caused by mainly three types of species. One is the Mycobacterium tuberculosis, another one is Mycobacterium africanum, and the third one is the Mycobacterium bovis. Out of these three, Mycobacterium tuberculosis is one of the most important bacteria which will cause the tuberculosis. And if you see the shape of the bacteria, these are straight or slightly curved rods. As they are in the rod shape, they are called as the bacilli. And these are gram positive bacteria, and they don't have cell membrane. They don't form any endospores or capsules. And here the important point you have to remember regarding this Mycobacterium tuberculosis is its cell wall is very thick and it is made up of the mycolic acids. Okay, its cell wall is very thick and is made up of the mycolic acids, and it is waxy in nature. As this is made up of the mycolic acids, these are very tough. The cell wall is very hard. Okay, and the next one, if you see the cultural characteristics, these bacteria will use amino acids and ammonia as nitrogen source and glycerol as a carbon source, and some bacteria are fastidious in nature, and they are mostly, these Mycobacterium species are obligately aerobes except Mycobacterium bovis, which is microaerophilic, and these bacteria are non-motile. The suitable temperature for their growth is 37 degrees centigrade and the pH is 6.4 to 7.0. And if you want to grow these bacteria in laboratory, the suitable media is Lowenstein-Jensen media without starch. Okay, so you have to remember this point also. And how the colonies will appear. So the colonies will appear as irregular, dry, raised rough colonies with milky white color. And if you observe in the microscope, they will appear as small clumps or pairs. And they don't get stained with stains easily, and they need some heating, and they are not at all stained with the gram staining. And we use Zeal-Nielsen technique. You know very well, this is also one of the very important point regarding this bacteria. Zeal-Nielsen technique we use for the staining of this bacteria, and uh, it don't absorb the stain easily until unless we can heat it. Later, if once it absorbs the stain, it don't lose that color even if you treat with the dilute acids also. That's why only it is called as the acid fast bacilli. And the next one is the pathogenesis. It is very important. So this tuberculosis spreads from one person to another person through the air when he coughs or when he spit or when he sneezes. Thus droplets or sputum, through the sputum and droplets, these bacteria enters into the air and they will enters into another healthy person. And if you see, mostly these bacteria which are entered into the lungs, they are killed by the macrophages of the upper respiratory tract only. If at all, if some bacteria are still alive, they will enters into the alveoli of the lungs, and they will start to divide and infection will start. Okay, and they will survive in the macrophages. They will survive in the macrophages, and the immunity which will act against these Mycobacterium tuberculosis is the cell mediated immunity. This is also one of the important point you have to remember. Cell mediated immunity will takes part in the immunity against the Mycobacterium tuberculosis. And whenever these bacteria enters into the macrophage, and it will stay there and it will divide, its number will increase, what it will happen, lesions will form. So lesions means తెలుగులో చెప్పాలి అని అంటే వాట్ పుండు పడుతుంది కదా అండి అలాగా. సో lesions will start. These characteristic lesions are called as the tubercle. This characteristic lesion is called as the tubercle, and it will starts to infect the tissue surrounding that. And these tubercles or lesions are two types. One is the exudative lesion and another one is the productive lesion. Exudative lesion will contains polymorphonuclear cells along with the edema fluid, and productive lesions contains only cells. And this tuberculosis disease is also two types. Okay, first one is called as the primary tuberculosis and second one is called as the post-primary tuberculosis. Primary tuberculosis. If you see the primary tuberculosis, this primary tuberculosis is caused by the entry of the pathogen, and usually we will see this in the young children. So whenever it enters into the lungs and it will stay in the macrophages and it will divide, it forms the lesions as we discussed earlier, and they are also called as the gone focus, and these lesions are formed within 3 to 8 weeks after the infection was started, and it is also associated with the tuberculin hypersensitivity. This is one of the important point, tuberculin hypersensitivity. Okay, and these lesions are healed with the calcified nodules within 2 to 6 weeks, and in this time, usually in some cases, automatically with the immunity power of the individual, it will get healed. In some cases by treatment also, it will be cured. So like this will happen, and after primary infection, and when all the existing cells are killed and all the lesions are cured, so it will be okay. But in some cases, when there is still some alive cells are there and which are active in stage, they will cause the second phase of the infection. It is called as the post primary tuberculosis. It is caused by the activated existing pathogens, and these activated pathogens, or the remaining pathogens, these will causes the again lesions, and these lesions leads to the necrosis of the tissue, and tissue destruction will be happened. And by this necrosis, this necroted material will comes through the sputum while coughing or sneezing from the patient, and it will enters into the air and it will reaches to the another person, which will carry the bacteria, and it will causes disease in the another person. So this is the pathogenesis of the tuberculosis. And if you see the symptoms of the tuberculosis, so cough is there for more than 3 weeks, and chest pain will be there, low grade fever, night sweats, loss of appetite and loss of weight. We will see all these symptoms. And there is one more chance is there that these bacteria infecting material or necroted material, it will enters into the bloodstream and reaches to the different kinds of body parts such as bones, kidney, brain, lymph nodes, or some areas. And this disease caused in the another body parts other than the lungs, this it is called as the extrapulmonary tuberculosis, and the symptoms are depends upon the infected area. Okay, and the specimen what we have to take for the diagnosis is the sputum, CSF, urine, pus, or body fluids. Sputum is common, and if there are any symptoms other than the lungs disease like extrapulmonary tuberculosis, we have to take the specimen according to that area. And if you see the laboratory diagnosis, the one is the tuberculin hypersensitivity test, and second one is the routine sputum examination, and PCR test and demonstration of, as I told you, hypersensitivity to tuberculin protein. So these are the things how we have to identify what the test we have to do and what the specimen we have to take. So this is all about the tuberculosis. Thank you very much. Thanks for watching.